students. Welcome back to BioDecode, where biology is decoded into simple steps. This is your trusted audio guide for learning biology the smart way. You're listening to the NCERT Class 11 Biology Audiobook, Unit 2, Structural Organization in Plants and Animals, Chapter 5, Morphology of Flowering Plants. Before we begin, if you haven't already listened to Chapter 4, Animal Kingdom and its Exercise Solution, it's available on our channel. That chapter explored how animals are classified based on their structure and evolution. Now, as we enter Unit 2, we shift our focus to the organization of living forms, starting with plants. In this chapter, we'll study the morphology of flowering plants, understanding how different external parts, such as roots, stems, leaves, flowers, and fruits, are structured and how these structures relate to their functions. You'll also learn the botanical terms used to describe plant form, modifications, and their adaptive significance in survival and reproduction. This audiobook is designed to make complex botanical terminology easier to understand through clear explanations and logical flow. Whether you're revising for exams, preparing for NEET, you can listen, learn, and visualize each concept effortlessly. So, plug in your earphones, focus your mind, and let's begin exploring the fascinating structure of flowering plants. This is BioDecode, and you're listening to Chapter 5, Morphology of Flowering Plants. Unit 2, Structural Organization in Plants and Animals. The description of the diverse forms of life on Earth was made only by observation, through naked eyes, or later, through magnifying lenses and microscopes. This description is mainly of gross structural features, both external and internal. In addition, observable and perceivable living phenomena were also recorded as part of this description. Before experimental biology, or more specifically, physiology, was established as a part of biology, naturalists described only biology. Hence, biology remained as a natural history for a long time. The description by itself was amazing in terms of detail. While the initial reaction of a student could be boredom, one should keep in mind that the detailed description was utilized in the later day reductionist biology, where living processes drew more attention from scientists than the description of life forms and their structure. Hence, this description became meaningful and helpful in framing research questions in physiology or evolutionary biology. In the following chapters of this unit, the structural organization of plants and animals, including the structural basis of physiological or behavioral phenomena, is described. For convenience, this description of morphological and anatomical features is presented separately for plants and animals. Catherine Esau, year 1898 to year 1997. Catherine Esau was born in Ukraine in 1898. She studied agriculture in Russia and Germany and received her doctorate in 1931 in the United States. She reported in her early publications that the curly top virus spreads through a plant via the food-conducting or phloem tissue. Dr. Esau's Plant Anatomy, published in 1954, took a dynamic developmental approach designed to enhance one's understanding of plant structure and an enormous impact worldwide, literally bringing about a revival of the discipline. The Anatomy of Seed Plants by Catherine Esau was published in 1960. It was referred to as Webster's of Plant Biology. It is encyclopedic. In 1957, she was elected to the National Academy of Sciences, becoming the sixth woman to receive that honor. In addition to this prestigious award, she received the National Medal of Science from President George Bush in 1989. When Catherine Esau died in the year 1997, Peter Raven, director of Anatomy and Morphology, Missouri Botanical Garden, remembered that she absolutely dominated the field of plant biology even at the age of 99. Chapter 5, Morphology of Flowering Plants. The wide range in the structure of higher plants will never fail to fascinate us. Even though the angiosperms show such a large diversity in external structure or morphology, they are all characterized by presence of roots, stems, leaves, flowers, and fruits. In chapters 2 and 3, we talked about classification of plants based on morphological and other characteristics. For any successful attempt at classification and at understanding any higher plant, or for that matter, any living organism, we need to know standard technical terms and standard definitions. We also need to know about the possible variations in different parts, found as adaptations of the plants to their environment. For example, adaptations to various habitats, for protection, climbing, storage, and so on. If you pull out any weed, you will see that all of them have roots, stems, and leaves. They may be bearing flowers and fruits. The underground part of the flowering plant is the root system, while the portion above the ground forms the shoot system. Figure 5.1. The roots. The majority of the dicotyledonous plants, direct elongation of the radicle leads to the formation of primary root, which grows inside the soil. It bears lateral roots of several orders that are referred to as secondary, tertiary, and so on roots. The primary roots and its branches constitute the taproot system, as seen in the mustard plant, figure 5.2a. In monocotyledonous plants, the primary root is short-lived and is replaced by a large number of roots. These roots originate from the base of the stem and constitute the fibrous root system, as seen in the wheat plant, figure 5.2b. In some plants, like grass, monstera, and the banyan tree, roots arise from parts of the plant other than the radicle and are called adventitious roots, figure 5.2c. The main functions of the root system are absorption of water and minerals from the soil, providing a proper anchorage to the plant parts, storing reserve food material, and synthesis of plant growth regulators. Figure 5.1. Parts of a flowering plant. This figure shows the complete structure of a flowering plant. You can see the root system anchoring the plant into the soil, absorbing water and minerals. The stem, upright and sturdy, supports leaves and flowers and acts as a transport channel for water, nutrients, and food. Emerging from the stem are leaves, which are the primary sites of photosynthesis. The apex of branches or axils, we see flowers responsible for reproduction. Some plants may also show fruits developing after fertilization. The figure highlights the interconnection of all these parts and illustrates how each contributes to the survival, growth, and reproduction of the plant. Figure 5.2. Different types of roots. This figure illustrates three main types of root systems. A taproot, a single, thick, main root growing vertically downward with smaller lateral roots branching off. Typical in dicots like carrot or radish, it helps in anchorage and storage of food. B. Fibrous root. Numerous roots of almost equal size arising from the base of the stem, forming a dense network. Seen in monocots like grass and wheat, it prevents soil erosion and efficiently absorbs water from the surface layers. C. Adventitious root. Roots rising from unusual parts of the plant, such as stems, branches, or leaves, rather than from the radicle. Examples include banyan and sweet potato, providing additional support or storage. Regions of the root. The root is covered at the apex by a thimble-like structure called the root cap, figure 5.3. It protects the tender apex of the root as it makes its way through the soil. A few millimeters above the root cap is the region of meristematic activity. The cells of this region are very small, thin-walled, and with dense protoplasm. They divide repeatedly. The cells proximal to this region undergo rapid elongation and enlargement and are responsible for the growth of the root in length. This region is called the region of elongation. The cells of the elongation zone gradually differentiate and mature. Hence, this zone proximal to region of elongation is called the region of maturation. From this region, some of the epidermal cells form very fine and delicate thread-like structures called root hairs. These root hairs absorb water and minerals from the soil. Figure 5.3. The regions of the root tip. This figure focuses on the tip of a root, divides it into distinct functional zones. From the very tip upward, root cap, protective structure covering the meristematic tissue, secreting mucilage to ease soil penetration. Region of meristematic activity, located just behind the root cap, where cells divide actively, responsible for root growth and length. Region of elongation, cells increase in size here, pushing the root tip further into the soil. Region of maturation, cells differentiate into specialized tissues, and root hairs develop here, increasing the surface area for absorption. This figure clearly demonstrates how a root grows and develops in an organized manner. The stem. What are the features that distinguish a stem from a root? The stem is the ascending part of the axis, bearing branches, leaves, flowers, and fruits. It develops from the plumule of the embryo of a germinating seed. The stem bears nodes and internodes. The region of the stem where leaves are born are called nodes, while internodes are the portions between two nodes. The stem bears buds, which may be terminal or axillary. Stem is generally green when young, and later often become woody and dark brown. The main function of the stem is spreading out branches, bearing leaves, flowers, and fruits. It conducts water, minerals, and photosynthesis. Some stems perform the function of storage of food, support, protection, and of vegetative propagation. The leaf. The leaf is a lateral, generally flattened structure born on the stem. It develops at the node and bears a bud in its axil. The axillary bud later develops into a branch. Leaves originate from shoot apical meristems and are arranged in an acropetal order. They are the most important vegetative organs for photosynthesis. A typical leaf consists of three main parts: leaf base, petiole, and lamina. Figure 5.4a. The leaf is attached to the stem by the leaf base and may bear two lateral, small leaf-like structures called stipules. In monocotyledons, the leaf base expands into a sheath covering the stem, partially or wholly. In some leguminous plants, the leaf base may become swollen, which is called the pulvinus. The petiole uphold the blade to light. Long, thin, flexible petioles allow leaf blades to flutter in wind, thereby cooling the leaf and bringing fresh air to leaf surface. The lamina, or the leaf blade, is the green expanded part of the leaf with veins and veinlets. There is usually a middle prominent vein, which is known as the midrib. Veins provide rigidity to the leaf blade and act as channels of transport for water, minerals, and food materials. The shape, margin, apex, surface, and extent of incision of lamina varies in different leaves. Figure 5.4. Structure of a leaf. This figure shows the anatomy and venation of a leaf. Parts of a leaf includes the lamina or blade, the flat surface that performs photosynthesis, petiole, a stalk connecting the blade to the stem, and sometimes stipules, small leaf-like appendages at the base. Reticulate venation, a network-like pattern of veins forming a web, typical of dicot leaves like hibiscus. Parallel venation. Veins run parallel along the length of the leaf, characteristic of monocots like grass. The figure illustrates how veins provide mechanical support, transport water and nutrients, and aid in photosynthesis. Types of leaves. A leaf is said to be simple when its lamina is entire or when incised, the incisions do not touch the midrib. When the incisions of the lamina reach up to the midrib, breaking it into a number of leaflets, the leaf is called compound. A bud is present in the axial of petiole in both simple and compound leaves, but not in the axial of leaflets of the compound leaf. Figure 5.5. Compound leaves. This figure shows two types of compound leaves, where a single leaf is divided into multiple leaflets. A pinnately compound leaf. Leaflets arranged along a central axis or rachis, resembling a feather. Examples include rose and neem. E. Palmately compound leaf. All leaflets radiate from a single point at the tip of the petiole, resembling the fingers of a hand. Examples include cotton and silk cotton. The figure emphasizes the difference between simple and compound leaves, and how such adaptations can increase photosynthetic efficiency or reduce damage from wind. The compound leaves may be of two types. Figure 5.5. In a pinnately compound leaf, a number of leaflets are present on a common axis, the rachis, which represents the midrib of the leaf as in neem. In palmately compound leaves, the leaflets are attached at a common point, that is, at the tip of petiole, as in silk cotton. Phyllotaxy. Phyllotaxy is the pattern of arrangement of leaves on the stem or branch. This is usually of three types: alternate, opposite, and whorled. Figure 5.6. In alternate type of phyllotaxy, a single leaf arises at each node in alternate manner, as in China rose, mustard, and sunflower plants. In opposite type, a pair of leaves arise at each node and lie opposite to each other, as in calotropis and guava plants. If more than two leaves arise at a node and form a whorl, it is called whorled, as in Alstonia. Figure 5.6. Different types of phyllotaxy. This figure illustrates how leaves are arranged on a stem, known as phyllotaxy, which helps in optimal light capture. A China rose, alternate. Only one leaf arises at each node alternately along the stem. This prevents shading of lower leaves. B. Guava, opposite. Two leaves arise at the same node, directly opposite each other. C. Alstonia, whorled. More than two leaves emerge from a single node, forming a circular arrangement. The figure demonstrates how leaf arrangement affects photosynthesis and plant architecture. The inflorescence. A flower is a modified shoot, wherein the shoot apical meristem changes to floral meristem. Inner nodes do not elongate, and the axis gets condensed. The apex produces different kinds of floral appendages laterally at successive nodes instead of leaves. When a shoot tip transforms into a flower, it is always solitary. The arrangement of flowers on the floral axis is termed as inflorescence. Depending on whether the apex gets developed into a flower or continues to grow, two major types of inflorescences are defined: racemose and cymose. In racemose type of inflorescences, the main axis continues to grow. The flowers are borne laterally in an acropetal succession. Figure 5.7. In cymose type of inflorescence, the main axis terminates in a flower, hence is limited in growth. The flowers are borne in a basipetal order. Figure 5.8. Figure 5.7. Racemose inflorescence. This figure shows racemose inflorescence, where the main axis continues to grow indefinitely. The flowers develop laterally in an acropetal sequence, meaning the oldest flowers are at the base and the youngest at the top. Examples include mustard and radish. This type allows continuous flowering and extended periods of pollination. Figure 5.8. Cymose inflorescence. This figure illustrates cymose inflorescence, where the main axis terminates in a flower, limiting further growth, and subsequent flowers develop from lateral buds. The oldest flowers are at the top or center, showing basipetal development. Examples include sunflower and hibiscus. This arrangement ensures a defined flowering pattern. The flower. The flower is the reproductive unit in the angiosperms. It is meant for sexual reproduction. A typical flower has four different kinds of whorls arranged successively on the swollen end of the stalk or pedicel, called thalamus or receptacle. These are calyx, corolla, androecium, and gynoecium. Calyx and corolla are accessory organs, while androecium and gynoecium are reproductive organs. In some flowers like lily, the calyx and corolla are not distinct and are termed as perianth. When a flower has both androecium and gynoecium, it is bisexual. Flower having either only stamens or only carpels is unisexual. In symmetry, the flower may be actinomorphic radial symmetry or zygomorphic bilateral symmetry. When a flower can be divided into two equal radial halves in any radial plane passing through the center, it is said to be actinomorphic. For example, mustard, datura, chili. When it can be divided into two similar halves only in one particular vertical plane, it is zygomorphic. For example, key, gomoher, bean, cassia. The flower is asymmetric irregular if it cannot be divided into two similar halves by any vertical plane passing through the center, as in canna. A flower may be trimerous, tetramerous, or pentamerous when the floral appendages are in multiple of 3, 4, or 5 respectively. Flowers with bracts-reduced leaf found at the base of the pedicel are called bracteate, and those without bracts, ebracteate. Figure 5.9. Position of floral parts on thalamus. This figure shows the relative positions of floral whorls on the thalamus receptacle. The hypogynous ovary is superior, and all other floral parts arise below it. B and C perigynous ovary is partially enclosed by a cup-like structure called hypanthium. Other parts appear at the rim. D epigynous ovary is inferior, with all other parts arising above it. The figure emphasizes the structural variation of flowers and their evolutionary significance in reproduction. Based on the position of calyx, corolla, and androecium in respect of the ovary on thalamus, the flowers are described as hypogynous, perigynous, and epigynous. Figure 5.9. In the hypogynous flower, the gynoecium occupies the highest position while the other parts are situated below it. The ovary in such flowers is said to be superior. For example, mustard, China rose, and brinjal. If gynoecium is situated in the center, and other parts of the flower are located on the rim of the thalamus almost at the same level, it is called perigynous. The ovary here is said to be half inferior. For example, plum, rose, peach. In epigynous flowers, the margin of thalamus grows upward, enclosing the ovary completely and getting fused with it. The other parts of flower arise above the ovary. Hence, the ovary is said to be inferior, as in flowers of guava and cucumber, and the ray florets of sunflower. Parts of a flower. Each flower normally has 4 floral whorls, viz., calyx, corolla, androecium, and gynoecium. Figure 5.10. Calyx. The calyx is the outermost whorl of the flower, and the members are called sepals. Generally, sepals are green, leaf-like, and protect the flower in the bud stage. The calyx may be gamosepalous, sepals united, or polysepalous, sepals free. Corolla. Corolla is composed of petals. Petals are usually brightly colored to attract insects for pollination. Like calyx, corolla may also be gamopetalous petals united or polypetalous petals free. The shape and color of corolla vary greatly in plants. Corolla may be tubular, bell-shaped, funnel-shaped, or wheel-shaped. Figure 5.10. Parts of a flower. This figure details the anatomy of a typical flower. It shows calyx sepals, protective green outer whorl, corolla petals, often colorful to attract pollinators, androecium stamens, male reproductive parts producing pollen, gynoecium carpels, female reproductive part with ovary, style, and stigma, pedicel, flower stalk connecting to stem. The figure provides a clear visual of how the flower is organized for sexual reproduction. Estivation, the mode of arrangement of sepals or petals in floral bud with respect to the other members of the same whorl, is known as estivation. The main types of estivation are valvate, twisted, himped, and vexillary. Figure 5.11. When sepals or petals in a whorl thus touch one another at the margin without overlapping, as in calotropis, it is said to be valvate. If one margin of the appendage overlaps that of the next one, and so on, as in China rose, lady's finger, and cotton, it is called twisted. If the margins of sepals or petals overlap one another but not in any particular direction, as in cassia and gulmohar, the estivation is called imbricate. In pea and bean flowers, there are 5 petals. The largest standard overlaps the 2 lateral petals' wings, which in turn overlap the 2 smallest anterior petals' keel. This type of estivation is known as vexillary or papilionaceous. Figure 5.11. Types of estivation in corolla. This figure shows how petals are arranged in the bud before blooming. Valvate. Petals meet edge to edge without overlapping. E. Twisted or contorted. Each petal overlaps the next in a spiral. C. Imbricate. Petals overlap irregularly like tile. D. Vexillary. Characteristic of legume flowers. One large standard petal overlaps 2 wing petals and 2 keel petals. This arrangement affects how flowers open and how pollinators interact with them. Androecium. Androecium is composed of stamens. Each stamen, which represents the male reproductive organ, consists of a stalk or a filament and an anther. Each anther is usually bilobed, and each lobe has 2 chambers. Pollen sacs. Pollen grains are produced in pollen sacs. The sterile stamen is called staminode. Stamens of flower may be united with other members, such as petals, or among themselves. When stamens are attached to the petals, they are epipetalous, as in brinjal, or epiphyllous when attached to the perianth, as in the flowers of lily. The stamens in flower may either remain free polyandrous, or may be united in varying degrees. The stamens may be united into one bunch or one bundle monodelphous, as in China rose, or 2 bundles diadelphous, as in pea, or into more than 2 bundles polyadelphous, as in citrus. There may be aeration in the length of filaments within a flower, as in salvia and mustard. Gynoecium. Gynoecium is the female reproductive part of the flower and is made up of 1 or more carpels. A carpel consists of 3 parts, namely stigma, style, and ovary. Ovary is the enlarged basal part, on which lies the elongated tube, the style. The style connects the ovary to the stigma. The stigma is usually at the tip of the style and is the receptive surface for pollen grains. Each ovary bears 1 or more ovules attached to a flattened, cushion-like placenta. When more than 1 carpel is present, they may be free, as in lotus and rose, and are called apocarpous. They are termed syncarpous when carpels are fused, as in mustard and tomato. After fertilization, the ovules develop into seeds, and the ovary matures into a fruit. Placentation. The arrangement of ovules within the ovary is known as placentation. Placentation are of different types, namely marginal, axile, parietal, basal, central, and free, central. Figure 5.12. In marginal placentation, the placenta forms a ridge along the ventral suture of the ovary, and the ovules are borne on this ridge, forming 2 rows, as in P. When the placenta is axial and the ovules are attached to it in a multilocular ovary, placentarian is said to be axile, as in China rose, tomato, and lemon. In parietal placentation, the ovules develop on the inner wall of the ovary or on peripheral part. Ovary is 1-chambered, but it becomes 2-chambered due to the formation of the false septum, for example, mustard and argemone. When the ovules are borne on central axis and septa are absent, as in dianthus and primrose, placentation is called precentral. In basal placentation, the placenta develops at the base of ovary and a single ovule is attached to it, as in sunflower, marigold. Figure 5.12. Types of placentation. This figure shows different arrangements of ovules inside the ovary. A marginal. ovules attached along the margin of a single carpel. B. Axile, ovules attached to a central column in a multicarpellary syncarpous ovary. C. Parietal, ovules attached to inner ovary walls. D. Free central, ovules attached to a central column with ovary walls free. E. Basal, a single ovule attached at the base. These arrangements are important for fertilization and seed development. The fruit. The fruit is a characteristic feature of the flowering plants. It is a mature or ripened ovary developed after fertilization. If a fruit is formed without fertilization of the ovary, it is called a parthenocarpic fruit. Generally, the fruit consists of a wall or pericarp and seeds. The pericarp may be dry or fleshy. When pericarp is thick and fleshy, it is differentiated into the outer epicarp, the middle mesocarp, and the inner endocarp. In mango and coconut, the fruit is known as a drupe, figure 5.13. They develop from monocarpellary superior ovaries and are one-seeded. In mango, the pericarp is well differentiated into an outer thin epicarp, a middle fleshy edible mesocarp, and an inner stony hard endocarp. In coconut, which is also a drupe, the mesocarp is fibrous. Figure 5.13. Parts of a fruit. This figure compares two fruits: a mango, fleshy, single-seeded fruit drupe showing exocarp, mesocarp, and endocarp layers. B. Coconut, a fibrous fruit drupe with a hard endocarp protecting the seed. The figure highlights the structural diversity of fruits and their adaptations for dispersal. The seed. The ovules after fertilization develop into seeds. A seed is made up of a seed coat and an embryo. The embryo is made up of a radicle, an embryonal axis, and one as in wheat, maize, or two cotyledons as in gram and pea. Structure of a dicotyledonous seed. The outermost covering of a seed is the seed coat. The seed coat has two layers, the outer testa and the inner tegmen. The hilum is a scar on the seed coat through which the developing seeds were attached to the fruit. Above the hilum is a small pore called the micropyle. Within the seed coat is the embryo, consisting of an embryonal axis and two cotyledons. The cotyledons are often fleshy and full of reserve food materials. At the two ends of the embryonal axis are present the radicle and the plumule. Figure 5.14. In some seeds, such as castor the endosperm, formed as a result of double fertilization, is a food-storing tissue and called endospermic seeds. In plants such as bean, gram, and pea, the endosperm is not present in mature seeds, and such seeds are called non-endospermous. Figure 5.14. Structure of dicotyledonous seed. This figure shows a dicot seed with two cotyledons, an embryo axis radicle and plumule, and seed coat. The endosperm is often absorbed into cotyledons. This structure supports initial growth after germination, providing stored food to the embryo. Structure of monocotyledonous seed. Generally, monocotyledonous seeds are endospermic, but some, as in orchids, are non-endospermic. In the seeds of cereals such as maize, the seed coat is membranous and generally fused with the fruit wall. The endosperm is bulky and stores food. The outer covering of endosperm separates the embryo by a proteinous layer called aileron layer. The embryo is small and situated in a groove at one end of the endosperm. It consists of one large and shield-shaped cotyledon known as scutellum and a short axis with a plumule and a radicle. Plumule and radicle are enclosed in sheaths, which are called coleoptile and coleorrhiza, respectively. Figure 5.15. Structure of monocotyledonous seed. This figure illustrates a monocot seed, such as maize, with a single cotyledon called the scutellum. It also shows endosperm, plumule, and radicle. Monocot seeds retain a significant endosperm as a food reserve for the developing seedling. Semi-technical description of a typical flowering plant. Various morphological features are used to describe a flowering plant. The description has to be brief, in a simple and scientific language, and presented in a proper sequence. Plant is described beginning with its habit, vegetative characters, roots, stem, and leaves, and then floral characters, inflorescence, and flower parts. After describing various parts of plant, a floral diagram and a floral formula are presented. The floral formula is represented by some symbols. In the floral formula, AR stands for bracteate, capital K stands for calyx, capital C for corolla, capital P for perianth, capital A for androecium, and capital G for gynoecium. G with line below indicates for superior ovary, and G with line above indicates for inferior ovary. A circle with a small arrow pointing diagonally upward to the right, like a spear, indicates male. A circle with a small cross extending downward, resembling a hand mirror, indicates female. A combination of male and female signs joined together indicates bisexual plants. A circle with a plus sign inside it indicates for actinomorphic, and percentage sign for zygomorphic nature of flower. Fusion is indicated by enclosing the figure within bracket, and adhesion by a line drawn above the symbols of the floral parts. A floral diagram provides information about the number of parts of a flower, their arrangement, and the relation they have with one another. Figure 5.16. The position of the mother axis with respect to the flower is represented by a dot on the top of the floral diagram. Calyx, corolla, androecium, and gynoecium are drawn in successive whorls, calyx being the outermost and the gynoecium being in the center. Floral formula also shows cohesion and adhesion within parts of whorls and between whorls. The floral diagram and floral formula in Figure 5.16 represents the mustard plant family Brassicaceae. Figure 5.16. Floral diagram with floral formula. This figure depicts a floral diagram, a schematic top view of a flower showing the number, arrangement, and fusion of floral parts. The accompanying floral formula summarizes the flower's features numerically and symbolically, indicating bisexuality, symmetry, number of sepals, petals, stamens, and carpels, and ovary position. It helps in easy comparison between different flowers. Solanaceae. It is a large family, commonly called as potato family. It is widely distributed in tropics, subtropics, and even temperate zones. Figure 5.17. Vegetative characters. Plants mostly herbs, shrubs, and rarely small trees. Stem. Herbaceous, rarely woody, aerial, erect, cylindrical, branched, solid or hollow, hairy or glabrous underground stem in Potato Solanum tuberosum. Leaves. Alternate, simple, rarely pinnately compound, stipulate, pinnation, reticulate. Figure 5.17. Solanum nigrum McCoy plant. This composite figure shows various parts of Solanum nigrum. Flowering twig. The stem with leaves and axillary flowers. B flower, bisexual, and actinomorphic. C. Longitudinal section of flower, showing ovary, style, and stamens. D. Stamens, male reproductive structures. E. Carpel, female reproductive structure with ovary, style, and stigma. F. Floral diagram, schematic view representing all the floral world. The figure illustrates both the macroscopic and microscopic floral structures in a single plant. Floral characters. Inflorescent, solitary, axillary, or cimose, as in Solanum. Flower, bisexual, actinomorphic. Calyx sepals 5, united, persistent, valvate estivation. Corolla, petals 5, united, valvate estivation. Androecium, stamens 5, bipetalous. Gynoecium, bicarpellary obligately placed. Syncarpus, ovary superior, bilocular, placenta swollen with many ovules. Oxyle. Fruits, berry or capsule. Seeds, many. endospermous. Therefore, the floral formula is read as combined symbol of male and female represents a bisexual or hermaphrodite flower. The circle with a plus sign inside it denotes an actinomorphic flower, and with 5 united sepals shown as capital K with 5 in brackets, 5 united petals shown as capital C with 5 in brackets, 5 stamens shown as capital A5, and a bicarpellary superior ovary shown as capital G with a line below and 2 in brackets. So, the complete floral formula for Solanaceae is symbol of bisexuality, symbol of actinomorphy, A5C5A5G with a line below 2225. Economic importance. Many plants belonging to this family are source of food: tomato, brinjal, potato, spice chili, medicine belladonna, ashwagandha, fumigatory tobacco, ornamentals petunia. Summary. Flowering plants exhibit enormous variation in shape, size, structure, mode of nutrition, lifespan, habit, and habitat. They have well developed root and shoot systems. Root system is either taproot or fibrous. Generally, dicotyledonous plants have taproots, while monocotyledonous plants have fibrous roots. The roots in some plants get modified for storage of food, mechanical support, and respiration. The shoot system is differentiated into stem, leaves, flowers, and fruits. The morphological features of stems, like the presence of nodes and internodes, multicellular hair, and positively phototropic nature, help to differentiate the stems from roots. Leaf is a lateral outgrowth of stem developed exogenously at the node. These are green in color to perform the function of photosynthesis. Leaves exhibit marked variations in their shape, size, margin, apex, and extent of incisions of leaf blade lamina. The flower is a modified shoot, meant for sexual reproduction. The flowers are arranged in different types of inflorescences. They exhibit enormous variation in structure, symmetry, position of ovary in relation to other parts, arrangement of petals, sepals, ovules, and so on. After fertilization, the ovary is modified into fruits and ovules into seeds. Seeds either may be monocotyledonous or dicotyledonous. They vary in shape, size, and period of viability. The floral characteristics form the basis of classification and identification of flowering plants. This can be illustrated through semi-technical descriptions of families. Hence, a flowering plant is described in a definite sequence by using scientific terms. The floral features are represented in the summarized form as floral diagrams and floral formula. You've just finished listening to Chapter 5, Morphology of Flowering Plants, from NCERT Class 11 Biology, here on BioDecode. I hope this audio helped you visualize and understand how each part of a flowering plant, from roots and stems to flowers and fruits, contributes to its form and function. We explored key botanical terms, various modifications, and the adaptive significance behind each structure. In the next episode, we'll go through the exercise solutions and important questions from this chapter, explaining each answer step by step to help you prepare confidently for your exams. So make sure to follow BioDecode and stay tuned for the next part of this series. Keep learning, keep exploring, and I'll meet you in the next audiobook to Chapter 5, Morphology of Flowering Plants, NCERT, Solutions and Key Questions. And yes, students, don't forget to like, share, and subscribe to our channel, BioDecode. See you in the next audio lecture.